Hello friends, welcome back to my channel Plant Pathology Pathsala. So in this video we will discuss about replication of viruses. So first we will discuss about replication of positive sense RNA virus, single stranded positive sense RNA virus. See, viruses may have single stranded or double stranded genome, that may be DNA or RNA. Okay. Again, this single stranded RNA virus, based on the genome polarity, it may be positive sense single stranded or negative sense single stranded. So basic difference between these single stranded RNA types is the positive sense single stranded RNA, it has same polarity with the cellular mRNA. So it directly acts as mRNA. So directly it will use the host ribosome to synthesize the proteins. It can be directly translated. But the negative negative sense single stranded RNA, it has not, it means its polarity is not similar with the mRNA. That's why it can be directly behave as mRNA. So this is the basic difference. So here we will discuss about replication of positive sense single stranded RNA viruses, how it multiply itself in the host cell. Okay, means multiplication of virus means we know that virus is a nucleoprotein structure. Nucleoprotein means nucleic acid will be there and protein coat is there. Okay, so to multiply itself, it has to produce more amount of nucleic acid and the coat protein. So these two things the virus need to be produced or need to synthesize. So by in this replication process, it will synthesize the coat protein and other associated protein and also produce or multiply its nucleic acid. Then they will assemble, finally they will form the nucleic acid that is and that enclosed inside the protein coat, and after the final assembly the progeny virus will be formed. Okay, so how this, because viruses have different type of genomes, so they have different strategy to multiply itself. So we will discuss one by one how these viruses are actually multiplying their genome or how they are producing their proteins. Okay, so first about the single stranded RNA virus. As you know that this positive sense single stranded RNA virus has same polarity as the cellular mRNA. Therefore, it can directly function as mRNA. Okay, means it will directly use the host machinery, the translational machinery to synthesize its protein. Okay, so let's discuss all the steps in detail. First, virus will first enter into the susceptible host. So entry of the single stranded virus may be either through the vector, that may be insect or nematode, anything. Okay, then or it may be due to the wound or through the mechanical transmission. Okay, so virus will enter, then after entering into the host, the viral capsid is removed. That process is called as uncoating. Okay, so the virus that capsid protein will be removed, and ultimately the genome will be entered into or released into the cytoplasm of the host cell or susceptible host. So here you can see, first it will attach, then enter into the host cell either through the wound or through the vector mediated or by mechanical transmission. Then uncoating will be there, means its protein or will be degraded, and then simply this nucleic acid will be released into the host cytoplasm. So as it has same polarity with the cellular mRNA, it directly acts as mRNA, and by the help of, by hijacking the host translational machinery, that is host ribosome and host RNA dependent RNA polymerase, it will uh, start translation. Okay, so how what it after translation, what it produce? So it produce some new enzymes like RNA dependent RNA polymerase, proteases, helicase, its code protein, and other associated protein. So virus actually first synthesize whatever uh, proteins it needed. First it will uh, synthesize the protein it needed. Okay, next step is formation of replication complex. See, virus can't only rely on the host enzymes, so it will first synthesize its own whatever enzymes require or whatever protein require. So major thing it produces the RNA dependent RNA polymerase. Okay, that is the central component. So now it will produce, actually the basic thing is virus is just a nucleoprotein structure. It doesn't have anything. So first, when it enter into the plant cell, it will hijack the plant translational machinery, a mechanism, and produce whatever proteins, enzymes it needed for its own replication. Okay, that are these proteins, proteases, helicase, and this RNA dependent RNA polymerase. This is the most important one. Okay, so now it will form the replication complex. So newly synthesized viral protein, particularly the RNA dependent RNA polymerase, associated viral RNA, host protein, intracellular membrane, they actually form a functional complex that is known as viral replication complex. It is nothing but just a complex of necessary proteins, enzymes that is required for replication or multiplication of plant viral genome, or for the multiplication of single stranded RNA virus. Okay, and the most important thing is RdRp, RNA dependent RNA polymerase. Why it is called as RNA dependent RNA polymerase? Because the template used for production or multiplication of RNA is a RNA only. So the single stranded RNA, this enzyme RNA dependent RNA polymerase use single stranded RNA for multiplication of or for polymerization of RNA. That's why it is known as RNA dependent RNA polymerase. Okay, so after the formation of this replication complex, it will start synthesizing the anti-genome or negative strand RNA, because when this only because when virus entered into the plant cell, its protein code is degraded, only we have our nucleic acid, that is a single stranded positive sense RNA genome, and some associated protein that we got after translation. Okay, and all this replication complex, RNA dependent RNA polymerase, this thing we have. So this RNA dependent RNA polymerase use this virus single stranded RNA genome as template and synthesize negative strand single stranded RNA. Because this for positive sense single stranded RNA, if we start polymerizing it, then definitely we will get the complementary strand. As it is a positive, definitely we will get the complementary negative strand. Okay, this is also known as anti-genome. So this is the This is the most important thing, the antigenome or the negative strand RNA. This is now act as template for multiplication of the positive strand RNA. See, from positive sense RNA, by the help of RNA-dependent RNA polymerase, we have synthesized this negative strand RNA, but it is of no use because our genome is of positive sense RNA. So we want positive sense RNA in more number. Okay, so now our RNA dependent RNA polymerase uses this antigenome for synthesis of positive sense RNA genome. So by the help of RdRp, they will form positive sense single stranded RNA. Okay, so what happened, these are complementary. Positive sense and negative sense RNA are complementary, that's why they will form bond, and that's why when a single stranded RNA start multiplying itself inside the host, we will get some replicative intermediate. These are nothing but the double stranded RNA, because the negative and positive strand that are synthesized are complementary to each other, so they will join and form this double stranded RNA that is known as replicative intermediate. Later they will separate, and this negative strand RNA are again used as template for production of more and more number of positive strand RNA, because the genome of the virus is positive strand. We don't need negative strand, only the negative strand RNA is used as template, okay, for the genome amplification. So now whatever newly synthesized positive strand RNA from the negative strand antigenome, what is the fate of that genome or nucleic acid? So it either undergo translation to produce the functional proteins like RdRp or structure code protein, movement protein, or other viral proteins and other viral protein that is required for the virus functioning. Okay, or it is for multiplying the genome of the virus, or ultimately they will assemble. So here the code protein and the genome will assemble and form the progeny virus. So in summary, what happened? Virus enter into the cell, its code protein is degraded, then the nucleic acid only enter into the cytoplasm, then the newly then this our nucleic acid or the single stranded RNA directly act as mRNA and start multiplication, start translation and produce some enzymes or replication complex proteins that are needed for the further replication of the RNA. And what is the fate of the RNA that produce or synthesize? Either it undergo translation or for genome replication, or for the progeny virus formation or encapsulation. Okay, this thing I have already told. First, virus will enter into the host cell. It will after entering the host only degrade its code protein. So code protein will be removed and only the single stranded RNA genome will be released into the cytoplasm. Then this single stranded genome will directly act as mRNA by hijacking its host ribosome. It start translation and produce some important proteins that is helpful for the replication, especially the RNA dependent RNA polymerase. Then with the help of RNA dependent RNA polymerase, the positive strand RNA will produce this intermediate RNA intermediate, that antigenome will be produced. So as these two are complementary strand, So they will join to form RNA intermediate. This is the characteristic thing for the replication of single stranded RNA. Now this negative strand or anti-genome is used as template by the RNA dependent RNA polymerase to form multiple number of single stranded positive sense genome. Now genome is already prepared. So what is the fate of that genome? Either it undergo further multiplication for producing more number of nucleic acid, or it produce some undergo translation and produce code protein, movement protein, and other virus associated proteins, and ultimately they will assemble. The nucleic acid will be encapsulated inside the protein code. Finally the virion, the progeny virion is ready. Either it transfer, or it move from one cell to another cell, or it move from means systematically spread through the vascular tissue to distant part. Okay, remember this movement protein, it facilitate the movement of virus from one cell to another cell via this plasmodesmata. This is for the short distance transfer. For long distance transfer, virus uses the phloem tissue. Okay, so this is about replication of positive sense single stranded RNA virus. Okay, then about the negative strand RNA genome we will discuss in the next video. Okay, now I think everybody is clear about the multiplication of positive strand RNA virus. Simply remember, positive sense RNA means it directly acts as mRNA, so it will translate its own protein. Virus will not carry any enzyme or anything. It will synthesize by hijacking the host machinery, host translation machinery, host ribosome, synthesize its enzyme and protein whatever it required for replication, and then it start replication by producing the major thing, that is the RNA dependent RNA polymerase. So by this enzyme it will produce the antigenome. Then antigenome will be act as template for further production of the main genome, that is positive strand RNA. And finally the code protein will be produced, new RNA are produced, and ultimately encapsulation will occur, new viral progeny will be formed. Okay, very simple. Okay, this is about positive sense RNA virus replication. Thank you for watching this video.